Some of you may be aware that there was actually a shutdown of the United States government. Did any of you know that? Yeah. All right, so this past week I've been working with the President, the House of Representatives. I called up Mitch McConnell because I told him that we had the director of CBER in the FDA that's supposed to be on a panel. We can't have a shutdown. So they listened, of course, to my call. And thanks to me, no, I am kidding you. Thanks to a miracle, we have our government functioning and funded, and we have the FDA in the House. I want to introduce Dr. Julie Allickson, the Director of Translational Medicine and Research at the Wake Forest Institute for Regenerative Medicine, to lead the next panel. Julie.
Good evening. Thank you. I'm happy to be here, and that last panel was fabulous, and I think it's really a springboard for the next session. And we really are in an era of change, and it's very exciting, and especially what's going on with FDA and all of the different pathways that they have in place now for us to be able to accelerate regenerative medicine. So just as Susan said, it's prime time now. So we're going to have a keynote speech, and then we're going to go into a panel. So I'd like to introduce, I'm honored and privileged to introduce Dr. Peter Marks. He is the Director of the Center of Biologics and Evaluation and Research at FDA. The center is responsible for assuring safety and effectiveness of biological products, as well as a lot of others. Dr. Marks received his medical degree at New York University. Following this, he completed an internal medicine residency in hematology, medical oncology, and a fellowship at Brigham and Women's in Boston, where he subsequently joined the attending staff as a clinician scientist and eventually served as the clinical director of hematology. He then moved on to work for several years in pharmaceutical industry on clinical development in hematology and oncology products, prior to returning to academic medicine at Yale University, where he led the adult leukemic service and served as the chief clinical officer of the Cancer Center. He joined the FDA in 2012 as deputy center director and became the center director for CBER in 2016. Dr. Marks is board certified in internal medicine, hematology, medical oncology, and is a fellow of the American College of Physicians. So honored and privileged to introduce Dr. Peter Marks. Where's the clicker? Who has for the slides?
Thank you.
Okay, great.
Thanks a lot. Wonderful. Thank you so much. Thank you so very much. Really, facilitating the development availability of safe and effective medical products for those in need is really one of the most important things that we do at FDA. And I really value the invitation to participate in this session today. I also want to thank the panelists. This last panel was really quite, I was quite inspired by it, because I think it really dovetails very nicely with some of the things that I will tell you. And in some ways, I think I don't have 5 words to say about it. I have 1 word: hallelujah. So we really are at a critical juncture in the field of regenerative medicine and stem cell biology. We're at a point now where advances in stem cell biology are really hitting the clinic. And I think these advances now can truly benefit those with a variety of conditions that really aren't addressed by conventional drug therapies. So really putting efforts into the thoughtful clinical assessment of these products so their potential can be realized, and so that there's really a way to get them reimbursed for people who otherwise are currently paying out of pocket for stem cell therapies, is really something that we would like to see happen. So what I'll try to do in the next 25 or 30 minutes is, in what I was thinking you might want to hear about, are a little bit of what the Center for Biologics is about, and a little bit of our history helps you understand how things get regulated or come to be regulated. I want to very briefly touch upon a description of the complex biologics that we regulate, prior to moving on to some examples of cutting-edge biologic products and cutting-edge cellular therapies, and then really spend the bulk of my time talking about our accelerated programs for regenerative medicine and kind of the framework by which we regulate regenerative medicine, and also talk about our efforts for facilitating development. So the Center for Biologics actually predates the Food and Drug Administration in terms of its formation. At the beginning of the 20th century, the two most common biologic products were diphtheria antitoxin and smallpox vaccine. And unfortunately, just after the turn of the 19th to 20th centuries, there were some very bad manufacturing events that occurred. In one case, unfortunately, some serum that should never have been released from a horse that was known to have had tetanus was released and ended up killing about a dozen kids. And there was a similar episode in a different city that led to a total of quite a number, about 22 children dying. And that actually led to the passage of the Biologics Control Act of 1902 under Republican administration of Teddy Roosevelt, and the formation of what was the precursor ultimately to the Center for Biologics Evaluation Research. The products that we regulate, though, now go from that vial of diphtheria antitoxin, which, by the way, is still a licensed biologic product, to the products that are coming in now under IND for CRISPR-Cas9 genome editing. And so really, over the past 100 years or so, plus of our history, we've evolved. We were part of the National Institutes of Health for part of our lifespan as the Center for Biologics, and only came to be part of FDA in 1972. The center has a very significant research component. We have about 300 scientists who conduct applied scientific research. They're not basic researchers. They look for ways to build better assays, build better ways to grow cells, to manufacture. We have about 1,200 full-time employees overall. We're now all consolidated at the White Oak campus. Until a few years ago, we were on the NIH campus and up and down various areas in Bethesda and Rockville, Maryland. So we've moved into one location, and that's been a big help. So the products that we regulate include a variety of complex biologic products. We don't regulate most therapeutic biologic proteins. Those are regulated by the Center for Drug Evaluation and Research. And if we had a half an hour, I'd tell you the politics about 15 years ago that led to that. But what we do regulate are the more complex biologic products, vaccines, allergenic products, live biotherapeutic products, blood products, a variety of devices related to biologics, human tissues and cellular products, to wit this conference, xenotransplantation products, and gene therapies. And this is really quite an interesting profile of products. Within this, when live biotherapeutic products, it's pretty amazing. There are things that we wouldn't have. When I came to the FDA, never thought that we would be regulating fecal microbiota transplant. In fact, I probably would have run the other way had they told me about it. But we regulate everything in our center, from stool to leeches to CRISPR-Cas9. So it's really kind of cool. What I want to spend a moment talking about is just something that's, I think, important for us to realize. And I think this is something that helps us understand why this field of regenerative medicine and cellular therapies is really only coming to be mature now. When you think about the complexity of therapeutics, small molecules, they're usually, when you have small molecules, we understand how to analyze them very well. Most of them can be cooked without losing activity. When you're done making them, If you haven't done a good job making them, you can figure it out usually by using chromatography or any one of a number of other analytic means. And even for therapeutic proteins today, one can generally figure out with monoclonal antibodies or peptide hormones whether you've made it correctly or not. And the analytics there are pretty good. But when you come to cells, what you're talking about now are combinations of tens of thousands of proteins, lipids, other molecules that come together. And you're actually talking about something that can actually react to its environment. And so I, as a cell biologist by training, I'm always very impressed that cells have their own personalities. And that means that when we're actually thinking about figuring out what we have when we have a cell product, it's incredibly more complex than when we have a small molecule or even a single protein. So really, what it's led to is the need to put in quality by design for cellular manufacturing, because at the end of the day, it's very hard to tell unless you've made it consistently by a reproducible process, whether you're making the same product over and over again. So this has been one of the challenges, and I think it's continuing to be one of the challenges that we'll all have to work through. So this is just to talk about our advanced medicinal therapy products. I mean, we really have some really products at the leading edge of medicine. We have ex vivo and in vivo gene therapies to treat various conditions. On the left side here with the ex vivo therapies, you see really cells removed from the body, altered in a cell culture, and then readministered after they've been modified. Tremendous potential there. And then the in vivo gene therapies, which really are just coming into being a reality. And then we have regenerative medicine products. And this really is a field of really great promise that includes a variety of innovative products from the cellular therapies, which, at least from our definition, includes genetically modified cells and gene therapies producing durable effects. It includes tissue engineering products, human cell and tissue products, and combination products that involve cells. And really, when you think about it, there's such an incredible diversity and wide array of products in this area that have the potential to meet so many different unmet medical needs, everything from bioengineered skin, bioengineered blood vessels, bioengineered organs, particularly simple organs like the bladder. Obviously, not all of these have seen the light of day in regulatory approvals yet, but one that has is the one at the bottom right here, the chimeric antigen receptor T cells. And here you see a cancer cell in yellow being attacked by a CAR T cell in red. So really an incredible array of products in development. And I just want to by way of looking at what success looks like, just spend a moment on looking at CAR T cells and what they mean. Sometimes success means being on the cover of a magazine. Other times that's probably not a good thing. These days, being on a magazine cover could be dubious or it could be an honor. But these really have been quite exciting. Chimeric antigen receptor T cells, many of you are probably aware of them, but these are a cell-based gene therapy that have potential applications to treat multiple different diseases, although the first ones that have become licensed products are treating hematologic malignancies. They may ultimately be able to treat some solid tumors, although there's a lot of investigation in that arena. They may be able to treat infectious diseases, and they're under investigation for autoimmune disease because the idea is the T cells, immune system, modulating immune system may be of benefit as well. And one of the great things about these is they have the potential to provide therapeutic benefit that has an extended duration of benefit. And the business end of these really has been facilitated by molecular genetics, being able to essentially make these chimeric molecules where this is just one embodiment where you have an antibody head that's been grafted to an activating or activatable T cell receptor, such that when the antibody head sees its target, it activates the T cell to both proliferate and to kill the target. So these really have potential for not just cancer, but for other avenues or diseases where you want to eliminate a given cell type. And so we see that right now there are two chimeric antigen receptor T cell products that have been approved. And it's just worthwhile just thinking about them. Kymriah, which was the first to be approved for patients up to 25 years of age with B-cell precursor acute lymphoblastic leukemia that's refractory to second or later relapse. This is really, as an adult oncologist, I spent some time treating kids on bone marrow transplant units. This has got to be one of the most awful things. Kids, because we now cure about 85 or 90% of kids with acute lymphoid leukemia. And the ones we can't, though, it's just so heartbreaking for their parents. So this is really addressing a tremendous unmet medical need. And the amazing thing with this particular therapy was in a place where there really was nothing effective, about 85% of kids went into a complete remission. So that's a tremendous effect. Didn't need a randomized trial to show that. So that's, I think, just to keep that in the back of your mind as we move forward, just talking about things. Yescarta, another example, this time a CAR T cell addressing an adult disease, relapsed refractory non-Hodgkin's lymphoma, large B-cell lymphoma, that's failed 2 or more lines of systemic therapy. Here, about half of adults responded, had a complete response. Again, something that you just wouldn't see otherwise, and I can say that as somebody who's treated a heck of a lot of non-Hodgkin's lymphoma. So really, what these both show, as I think, are examples of therapies where large clinical benefits can lead to relatively quick regulatory approvals. But one of the important things they also illustrate is that over the past 25 years, Congress has given FDA an increasing number of pathways or programs to use to help expedite the development of important medical products that meet unmet medical needs. And what I want to do now is try to tell you a bit more about those after I essentially, and then tell you a little bit about how the regenerative medicine and stem cells fit into that. So really, we now have a number of these pathways or programs to use. We have fast track designation, priority review, which gets people a quicker review clock through the agency, 6 months instead of the 10 months. We have the ability to use accelerated approval, which allows us to use surrogate endpoints. And we have breakthrough designation, which Congress put into place in 2012 as part of FDASIA, which really, I think, did one of the most important things that we could do here for development, which was to actually increase our conversation with those developing products. Granted, it also gives people potentially gives people all the other benefits here, fast track, priority review, accelerated approval, but it gives people a dialogue with the agency, and it also gives them the involvement of senior management in product development. It also helps people stock price. No, but that's just an aside. And then in the past, you can see I'm not a good comedian, so I'm going to stop trying to be one. Then in the past, about a little bit over a year ago, we had the Regenerative Medicine Advanced Therapies designation, which was already mentioned here, and I'll tell you more about that. That was part of the 21st Century Cures Act. I just want to back up for a moment and explain a little bit about how kind of this is a little bit dry, but it's kind of the framework by which we regulate human cells, tissues, and cellular and tissue-based products. So I'm going to abbreviate those HCTPs because it's a mouthful otherwise to say what these products are. So really there are two two portions of the Public Health Service Act that apply to the regulation of HCTPs. One is the section of the Public Health Service Act, Section 351, which allows us to license biological products. And for those products, that means we issue a license for distribution into interstate commerce. It holds the standards. These are the kind of the standards that were put in place, believe it or not, over 100 years ago, that products have to be safe, pure, potent, and it gives us suspension, revocation power, recall authority over those products. The other piece that applies to, that can apply to biologic products is Section 361 of the Public Health Service Act, and that gives us the authority to help prevent transmission of communicable diseases. And so really, these are the kind of the two laws we use. Believe it or not, there's also, obviously, the Food, Drug, and Cosmetic Act also applies to products, but this is what I'd like to just focus our discussion on, because when the regulation of tissues was thought about, it was realized that not everything needed to be a licensed product, because some things were essentially safe and were efficacious on their face value. And so two regulatory tiers were developed for HCTPs. One was the regulatory tier for ones that were considered drug devices or biological products, which we often call 351 HCTPs. And those are regulated under both the authority of Sections 361 and 351 of the Public Health Service Act, and also potentially also under the Food, Drug, and Cosmetic Act. And then there are 361 HCTPs, which meet certain criteria which allow them to be kicked down. In other words, they don't require a biologics licensing application. We just really allow for registration and listing of the facilities that produce them, and they are regulated solely under the provisions to prevent communicable disease transmission. And we have a set of tissue regulations as part of the Code of Federal Regulations under Part 1271. The most important thing is that pre-market review and approval aren't required. Now, obviously, when you have two regulatory tiers, and this is a visual effect, the burden for one set is really high, and the burden for another set is really, really low. You're going to have tension between them, particularly for products that fall on the line or close to the line. And so this has been a challenge over the past years, and it's really something that we realized needed additional guidance out there. And it needed additional guidance in part because the regulations for human tissues have been actually out there, final in form, since 2005. So to be regulated as these 361 HCTPs, there are several criteria that must be met. They have to be minimally manipulated. They're for homologous use only, not combined with another article, with some exceptions, and they generally don't have systemic effects. Or if they have a systemic effects, they're really in the related allogeneic transplant setting. And the kinds of products that are HCTPs are things like musculoskeletal tissue, cardiovascular tissue, skin, dura mater. And things that aren't HCTPs are vascularized organs for transplant, human milk, collagen. Importantly, not blood products either. So when we recently set forth the suite of regenerative medicine guidance documents, it was to try to help people get some clarity sorting their way through all of these terms, because I can barely say minimum manipulation without messing up both words. It is pretty confusing working your way through some of this. And so we wanted to clarify existing regulations to make it simpler for sponsors to determine if they needed to obtain pre-market authorization for our products, and also to expedite the development and approval of safe and effective innovative regenerative medicine therapies and associated devices. So this suite of guidance documents included 4 guidance documents, 2 of which are in final form, 2 of which are in draft form. Draft simply means that there's a comment period that's actually currently open. So if you want to say something to us about it, you can go to the federal docket system and enter a public comment. and we will see it. So the two final guidances are the same surgical procedure exception and the regulatory considerations for HCTPs, minimal manipulation and homologous use guidance. And the draft ones are one that was required by the 21st Century Cures Amendment on the evaluation of devices used with regenerative medicine advanced therapies, and one on the expedited programs for regenerative medicine advanced therapies. The same surgical exception guidance finalized a draft of pretty much the same name, and basically the same surgical procedure exception it tells people what procedures you don't have to involve FDA in because they're considered the same surgical procedure, and provided you stay within certain limits of processing steps, we will not need to be involved in any way in those procedures. And when are these, and it's not just the same surgical procedure. Some people think that it's in the same surgical suite. This actually comes up in neurosurgery because sometimes bone flaps need to be removed from the skull. That's just one example. The bone flap stays out for a few weeks. It then gets put back after a subdural hematoma or some other neurologic issue is addressed. And that meets this type of same surgical procedure exception. There are other things as well that may. So that's what this helps explain, what falls under that exception. The minimum manipulation homologous use guidance is probably one of the meatiest of these guidances, and that helps provide recommendations for how we apply the criteria of what is an HCTP on minimum manipulation and homologous use. And it supersedes a variety of other documents that were out there, both in draft form and final form. Really, it helps define what minimum manipulation means, what can be done and still be minimum manipulation, and what homologous use is by our criteria. And it has a lot of examples. It also has a flow chart in that guidance that helps people understand kind of how to figure out whether one needs a biologics license or not. One of the things that, and I'm sorry, I put the slide up a little in advance, and I think I got people distracted on this, but one of the things that became clear when we did this is that some people's impression of what their product was, even people who were very earnestly developing what they had, may have flipped from being a product that they thought didn't require a biologics license application to one that needed one. And for people who were bona fide, developing bona fide products, we didn't want to suddenly create criminals. And so we actually have an enforcement policy and compliance policy that we've put out. And what that means is for the first 3 years after November of 2017, when this policy went into effect, for certain products, there will be a 36-month period of enforcement discretion based upon a determination of risk to public health. What that means is that for products that are determined to be at low risk of causing harm, we will have a 36-month period where people can come into the agency, have a dialogue about their products, figure out if their product really needs a biologics licensing application or not, and if so, come in with one and get that in place. There are certain places, though, that probably, I have to say, we will take enforcement action, and it was already alluded to, we have taken enforcement action when products are actually harming people. There are cases where it's been pretty clear, publications in the New England Journal, a host of reports that we get, but we can't share, again, but I can tell you they're out there where people have been harmed by stem cell therapies. We will take enforcement action because people should not be harmed by therapies, particularly ones that they're paying for as patient-sponsored research. So we will take action against those. Those are mostly going to be ones that are administered by intravenous injection or into the central nervous system. And we'll prioritize those over ones where there's a lower risk. In addition, in general, when these therapies are intended for non-homologous use, we will tend to take enforcement actions more often in those conditions, again, because when someone is trying to treat autism using stem cells without a good IND in place, without a good rationale, when the patient's paying for it, and when there have been adverse events, that's just a hypothetical example, because we don't talk about compliance things unless we've taken action. This is where we would tend to go. Onto a more pleasant note, we have a guidance for regenerative medicine devices. That's really there to help people understand that we will try to apply the device, the device guidances that are in the Center for Devices and Radiologic Health, and that same principles of least burdensome to these devices as well. We will try to only use the highest risk classification, Class 3 devices, when it's absolutely necessary. And then the one that is probably most exciting to me in some ways is our expedited programs for regenerative medicine therapies. It's a draft guidance which describes our considerations in implementing the regenerative medicine advanced therapy designation as described in section 3033 of the 21st Century Cures Act. So really, the whole point of this is to expedite the development and review of regenerative medicine advanced therapies. It applies to certain cell therapies, therapeutic tissue engineering products, human cell and tissue products, and combination products. And it also, as I've mentioned, we apply this designation to genetically modified cell therapies and gene therapy products producing durable effects. And obviously, one can always argue about what durable means, but we're happy to have that conversation. The products in this program should be intended for serious or life-threatening diseases or conditions. There should be some preliminary clinical evidence that indicates the potential to address unmet medical needs. That's different from the breakthrough standard. The breakthrough standard says it has to have some advantage over existing therapies. So this is a standard that's a little bit easier to meet. We reply to designation requests within 60 days, and designated products are eligible as appropriate for priority review and accelerated approval. There's also some benefits on the post-approval commitment end of things, where we have this somewhat expanded group of things that can fulfill post-marketing commitments for accelerated approval, including use of real-world evidence or larger data sets from populations that have been studied. What I'd like to kind of conclude with is talking about what we have articulated in this guidance is hopefully something that will be of benefit to academic investigators and others. We may have some discussion. It's kind of a nice way, I think, to transition into the panel discussion, because there may be some discussion of this. We think that one of the ways that academics can work together and potentially even small companies is to have kind of an innovative development pathway where somebody comes up with a collaborative agreement to work together. Individual facilities that are using relatively simpler manufacturing processes that they can coordinate between different sites so that each individual site is making the product in a similar manner, can then perform clinical trials with patients at their own sites, and then can bin those data together for submission for a biologics license application, and then each individual site can have a biologics license. That way, although you start off alone, you join with others, and at the end of the day, you're not always beholden to everyone else because the license is your own. And so it's a little bit different from the traditional development of a biologic product, which is kind of shown on the left here. However, I think it really does have the potential to help those forward. And there have been some pilot programs in this area that I think are promising. Obviously, it requires a collaborative approach. It requires some lawyers to be involved to make sure there are good agreements in place. But we think it does have the potential to help facilitate things, because that's really what we'd like to see happen in this field. Really, at the end of the day, let's see, can we go back a slide? Can someone take me back a slide? What I what I really wanted to say to conclude is that we really welcome the opportunity to have a meaningful dialogue with people who are developing products. I think we'd really like to go away from looking like the big bad wolf to more, and we may not look exactly like grandma, but we like to be able to have a dialogue with people, because what we find is when people come in early and have a dialogue with us, not only do they realize that we actually can provide useful suggestions and that we're somewhat reasonable, they also sometimes find that we will help them avoid going through some things that they might have spent money on or spent time on that they actually don't need to. So we really encourage people to contact us. We have a particular type of meeting that's called a pre-IND meeting. I should probably have a naming contest for it because it sounds so similar to pre-IND meeting. That's a formal meeting, but a pre-IND meeting is an informal type of meeting that we have where basically you send us a page or 2 or 3 on what your product is and a few questions about what you'd like to talk about, and we can arrange a teleconference or an in-person meeting to help discuss how to move product development forward. And also here, there's additional information that can be found on our website, or people can contact us at industry.biologics@fda.hhs.gov. Really, ultimately, just to conclude, I really value the ability to be here with you because I think we share with you the desire to see these therapies take prime time. I think we understand that in the past, perhaps FDA has not always been as great at facilitating development as people might like us to be, but as I say, we'd like to become, again, we may not look like grandma, but we certainly are not the big bad wolf. So I look forward to our panel discussion. Thanks very much. Sure.
Are there any questions that are specifically related to this presentation? Otherwise, we can move into the panel if somebody has something specific. Anyone? Oh, yes, yeah.
Yeah, I was curious before you mentioned that blood products will not be considered to be covered, but what about umbilical cord blood?
So umbilical cord blood is umbilical cord stem cells is what you'd be talking about, and so those yeah. So there, when I'm talking about blood products, we're essentially platelets, plasma, red blood cells are not HCTPs. And I think actually the last of those that one of them I mentioned, platelet-rich plasma is something that orthopedists use a lot, and it's a lot of orthopedists thought they needed perhaps BLAs for those, and depending on things, it's a different regulatory pathway. So they have concerns. They need to just come and talk to us.
So I have a question about like how exclusivity be handled when you talked about your different paradigm for like banding together to be able to share clinical data, the clinical manufacturing sites and so forth. Will be the first group that comes together? Will they get any exclusivity, or how is that even going to enter into it? Because that is a concern, I think.
Yep. So it's a very good point. And the thing that seems to be lucky in this case is that most of the times biologic products, even if they're to treat the same disease, even if they're made by similar methods, are usually different enough that it's not an issue here. Now, I guess at some point we'll probably have to deal with biosimilars in this area, but that'll bring a whole nother realm to it. But I think the hope is that just by the fact that biologics are, just so you understand, from the agency's perspective, if you have the same gene insert but a different vector, those are different biologic products. Same thing if you have the same, so basically there are lots of different ways. Biologic products are highly individual, like everyone in this room.
Just another quick question. In one of the guidance documents that I had read, it mentioned that cell selection was an example of minimal manipulation. Can you expand upon that? Because I feel like that's a pretty broad area.
So I think what you're trying to say is that depending on how you do your cell selection, There are some methods of cell selection that may or may not qualify as minimum manipulation, and I think this is one of those times when I would say if you have a concern about whether the method you're using triggers something more than minimum manipulation or not, I would say the tissue reference group, which you can find on our website, they are an informal way of, again, you send informal way of contacting us, and they will send you back an opinion about whether what you're doing triggers or not. And so people should take advantage of those. We really try to give a lot of thought to that. The tissue reference group is an informal way. It's not there. Any advice you get from them is not binding. You can, if you disagree with it, you can do what you want. If you submit a request for designation of something, that's a different story. That is a legally binding thing. But the tissue reference group is there to help for those kind of answers. There's a question back there. Maybe one more. So I think what you're saying is, why are we? I think we're not concerned. Intravenous use of administration of hematopoietic stem cells has been done so many times, I think, to date now, and has a safety record that is established. I think I'm not sure what you're trying to compare it to, but it's different, perhaps. We consider that fundamentally different than perhaps the intravenous administration of adipose cells, adipose-derived cells. It's just they're in essence all of our for reasons that I think we can come up with some ideas on. Our bone marrow cells always go for a ride in our blood every once in a while, so they're always doing that. So in some ways, bone marrow cells and blood cells are somewhat of the same ilk. So we don't get too excited about that, as opposed to when you take something like a mesenchymal cell or a cell that normally would not be circulating in blood and put it there. Great.
Thank you. So if you would join our panel, and would the panel members please come up? And if you have other questions specifically for Dr. Marks, you could ask at the end of the session as well. So I just wanted to give our panel a little bit of time to speak. So as they're sitting down, I just want to say is that we've got a lot of experience up here. I'm really excited to have this wealth of information. We have Dr. Tony Atala, from the director of Wake Forest Institute for Regenerative Medicine. He's a practicing surgeon and researcher in the area, and he was very instrumental in the first laboratory tissue engineered organ that got implanted into patients, and has been involved in several different products and clinical studies and clinical treatments of patients. And he has many types of awards and applications, and so many to mention. If you've ever been in his office, you could see all 4 walls filled with awards. So very impressive. And Dr. Camillo Ricordi is the Director and Chief Academic Officer at the University of Miami Diabetes Research Institute, and a distinguished professor of medicine for biomedical engineering and surgery and microbiology and immunology. And he has served as the responsible head for the NIH-funded GMP Human Process Development Facility at the Institute, and he has really led peers in leading scientists with the diabetes-focused research, as well as developing the device, the Ricordi chamber, that we use to isolate islets today. And he's led the team that performed the very first successful transplant with a bioengineered endocrine pancreas implanted as a 3D scaffold. So he's had a lot of experience in the field, and we look to him as a leader in diabetes. Dr. Arnold Caplan, is the director of the Skeletal Research Center and professor for departments of biology and oncology, pathology, biomedical engineering at Case Western Reserve, and he's a national and international scholar focusing on experimentation in the area of musculoskeletal and skin development. He's published a lot in those areas. He's done a lot of research with aging and cartilage and bone, skin, and other mesenchymal tissues, and he's really known as the father of mesenchymal stem cells, or medicinal signaling cells. So very honored to have the group of you. And so I'd really like to start it out with, we got a lot of clarity from Dr. Marks, so I'm really excited about that, and I think that helps everybody to understand where are my products going and what can I really do with them to accelerate it, because I do feel like it's a changing time as we see some of these other therapies, immunotherapies, moving to phase 2 and then licensure. So it's very exciting. We also see in the field that a lot of our clinical trials are in phase 2. 5 years ago, I would have told you they're all in phase 1. Now they're in phase 2, and we're seeing phase 3. So it's very exciting, and we know that if we have a wildly successful therapy that's safe, we potentially could be able to move it into licensure at phase 2. So very exciting, but we also know that there's a lot of challenges in the field. And so I'd really like all the panelists to discuss a little bit about a challenge or something that we could change in the field in the regenerative medicine space. So, Dr. Ricordi, would you like to start out with that?
Sure. Maybe it's a little an anomaly compared to other cell products we are discussing here, but I would like to make the case for the most severe cases of type 1 diabetes that we are addressing with islet transplantation, and a challenge that we have currently in the field. If I can state the problem, like we have 430 plus million of patients with diabetes worldwide and 5 million deaths a year, or 1 every 7 seconds. But for type 1 diabetes in the US, we have around 1.2 million patients, of which 30% are affected by the so-called unawareness of hypoglycemia, so they cannot sense when blood glucose drops below a certain level, and this would make them around 360,000 patients. Of these, 66% of those have recurring severe hypoglycemic episodes requiring assistance or hospitalization or an emergency intervention. That would make like 237,000 around approximate patients. And if you try all kind of technology intervention, educational blood glucose monitoring, pumps with the shutoff, you still have less than 30% of these patients that do not improve despite all possible attempts, which would make around 70,000 patients in the US. For those patients, there is not an alternative. An islet transplantation or transplantation could be considered a life-saving procedure. Now, as we heard, like over 10 years ago, FDA started regulating islet allotransplants as drugs, as pharmaceuticals, with all the details that we heard in the brilliant presentation that preceded me. But, and we diligently complied with these guidelines and started a phase 3 trial with FDA phase 3 trial sponsored by NIH, multi-center trial that lasted 10 years, over 100 million spent, and was a massive effort of going through standardizing manufacturing across 8 manufacturing facilities. I will not go into detail, we don't have time. But moral of the story, we now completed a successful phase 3 trial, met all the primary endpoint, submitted thousands of pages of manufacturing report, clinical report to FDA. But now we have a challenge that the rest of the world in these 10 years move from regulating islets from a pharmaceutical to regulate them like an organ transplant, because they say it's too different. Each donor is different. You have age, cause of death, manufacturing, and is practically a microorgan that you transplant. So like bone marrow transplantation, also they don't regulate islet transplant. So the next challenge that we have now in the United States is that while the rest of the world, Canada, England, France, Italy, Switzerland, Australia, are passing us, leaving us in the dust, treating their patients. Edmonton passed 600 patients transplanted. We did 2 in Miami last year. The cost is unbearable, and the challenge is that now we were supposed to submit a BLA application from each center of the multi-center trial, so it would make it, nobody wants to fund this because they say, well, you're not a company, will not do it because it's a non-profit procedure. There is no patent because I made the mistake to freely make the patent available and renounce to any kind of royalty or benefit from it. So say, why shall we give you $2 million to do a BLA from which there is no return on the investment? So it's a little challenge in this direction, and I think maybe with these new guidelines and facilitated approval, we had some very creative exchanges and very beautiful interaction also with Bruce Schneider at CBER, and say maybe there is a way that we can rethink a facilitative path for approval of islet cell transplant so that also our patients in the United States can benefit for a procedure that sometime could be considered life-saving.
Does anyone on the panel have a comment?
Yeah, I mean, obviously, I'm very happy to comment, which is, I'm not going to comment directly, but I'll say this, that, you know, clearly this is an area where I think there's a tremendous, it's a great example of a tremendous clinical benefit, which is apparent. It is true that there are different regulatory structures for cellular therapies across different places. I happen to think that there's hopefully a way forward here where under our regulatory authorities, it can be, these therapies can come to benefit of people without being overly burdensome. That being said, I'll answer the second part of your question, the first part of the question that you asked, which is that, you know, from our perspective, one of the most challenging parts in a lot of these things has been the manufacturing piece, and I think that's been the most challenging piece particularly for academic investigators to get together. And to the extent that, that we identified this as a challenge, this is something that I think we're going to be talking about more with our colleagues at NIH about finding ways that we can help us. This is where I think that the field needs help facilitating their ability to submit manufacturing data that meets the requirements. Sometimes the manufacturing has been done correctly, it just hasn't been documented correctly. And so we'd like to not have that be the holdup for important therapies getting to people.
Thank you. Thank you. So I will add the tools are very important, and as we look at manufacturing, there are a lot of challenges in manufacturing, but automation hopefully is on its way, and that there's a lot of groups, including the RIMS that is going to be meeting on Friday that's going to be talking about automation and manufacturing. So it definitely is a hot topic, and we're all looking at how it can move forward. So thank you. Dr. Atala, would you like to address a challenge that you're seeing in the field?
Yeah, I think one of the major challenges that I see in the field is actually the cost of the production of these products. It's extremely high. We're talking about biologics, we're talking about autologous therapies, we're talking about homologous therapies. But you know, just the actual regulatory path, in fact, towards production of these products in terms of accurate manufacturing, like Dr. Mark just mentioned, these are very expensive reagents, very expensive processes and procedures. And actually, I'm surprised that we've come so far along in the field and we're still doing things the way we used to do them, you know, 50 years ago. And so how can we actually improve the costs and lower the cost by automation, like has been mentioned, but also by perhaps trying to standardize some of these processes and procedures that have gone through the approval process time and again, but unfortunately you don't necessarily have access to that information. And how do we make that change for the future as we move forward? In a perfect world, we would all be sharing that information with each other so we can help each other out with our products.
Does anyone have any comments on that? I would just say that I'm hoping that we're going to all be able to find a pre-competitive space where there are a lot of partnerships and we can move forward, and then at the end of the day there is exclusivity and there's IP and, you know, things that we can share in the beginning, but yet that there's a return on investment at the end. So that's my dream.
No, I would say that I think we're very hopeful, and I think we're trying to put a lot of effort into supporting efforts by those who are trying to help come up with ways of manufacturing. Exactly as you say, standardization. It would be ideal in some cases to have platforms or almost cassette-based cellular manufacturing that would let people produce products in a more cost-effective way, because I think you're right, people are we get to see, because we get to see everyone's stuff, we get to see people reinvent things over and over, whereas I think if people were exchanging information in a pre-competitive space, the whole field would end up further along. There is some good news in that There are some public-private partnerships involving, and some involving academic institutions too, NIMBLE and Army in this space that hopefully will bring things forward. I know the National Science Foundation as well is trying to help move forward manufacturing so that really a rising tide floats all boats, so to speak.
Thank you. Dr. Caplan, would you like to talk about?
I'd like to make 2 points. One is in the late 1980s, we invented mesenchymal stem cells, and we then 3 years later published a standardized procedure for identifying batches of fetal calf serum that would optimally support MSC proliferation and for differentiation. And there are currently thousands of laboratories who are using MSC technology and publishing thousands of papers. And it's exactly the point that Dr. Atala made. Nobody's doing it the same way. And so if Lonza or some large manufacturer could provide at reasonable costs aliquots of mesenchymal stem cells as a standardized procedure, instead of giving 10,000 doses to Mesoblast, give them to us and let us do our science with a standardized single donor batch of MSCs, it would change the course of MSC science. So that's soapbox one. Soapbox two, I can't resist, it's killing me, but I can't resist. Dr. Marks is sitting next to me, so I want to ask a hard question. But I'd like the audience also to participate by opening your minds and thinking about clinical trials. So I know a lot about MSC clinical trials. And so I would say MSCs have a fabulous safety profile. And what we know from the published data of Mesoblast and others is that by phase 2, we identify 50 to 60% of the patients treated have a non-responder profile. So can we use this is going to kill you, Peter. Can we use the non-responder profile instead of placebo controls? Because they are, in essence, the best control we could possibly have and identify a placebo effect. If in phase 1, we show a clear differential between placebo and treated, right? And we have a long safety profile. Please, you don't have to answer it here in public, but it's just we need in this industry a way of changing logics for cell-based therapy. We can't be guided by pharmaceutical logics because, as you pointed out, this is not a pharmaceutical. We can't do a mass spec of cells and guarantee their purity and efficacy. So let's change our logic. Recruiting patients and telling them they're the placebo control, you lucky dog, ain't going to work for us because we can only do clinical trials with 100 patients, not with 5,000 where you hide the non-responders. We want to count the non-responders and actually then stop doing placebo controls, because that will give us a realistic baseline on which we can move forward. And I think those kinds of uniform suggestions across the industry would help every single corporate entity and would help us who are trying to do investigator-initiated trials, because if we have 100 patients and 33 of them are placebo controls, enrollment is deadly. If we tell every patient, you're going to get the product, but there's a 50-50 chance it's not going to work, that's called consenting. And we can use those procedures. So I would just challenge the physicians in the audience and challenge the people in industry and challenge the people in the regulatory network. We need to change our logics. And I think changes of logics are going to be driven also by the economics of the products that we're dealing with. We can do a 100-patient trial. We can get statistics. But we need to make sure that the FDA allows us to, with appropriate surrogate assays, subtract the non-responders. Because in all of the published trials that I've looked at, by 3 months we can identify the non-responders. Mesoblast has 3-year outcomes on low back pain. But it's only with the 40% that responded at 3 months. And so again, my view, this industry in some ways is moving forward. And again, the 21st Century Cures Act was a major leap forward in giving the FDA the legal basis for changing lots of the rules and regulations. And one of them is a more serious consideration of the placebo control. That would be my other soapbox as a challenge.
Yeah, so I think we're very open to novel trial designs, and I think coming soon in some venue, you might see us talk more about this idea of how you potentially build off of trials where you have a non-responder population. It may even be by using that non-responder population for a period of time as your comparison population. And I think that may, if you have a real effect, I mean, that really what we're looking for here is ways of documenting a real effect of therapies. And I think we realize that that's going to be the case not just with cellular therapies, but truthfully, it's the case with even some targeted therapies and immunotherapies. It's going to be the same exact thing, that if we may find ways where you have documented non-responders that can then serve the function of a control group. So we're very open to those conversations. And I think more so than anything else, I think we are acutely aware of the fact that we have to find ways of being able to conduct clinical trials in this area with reasonable numbers of patients, because otherwise we're doing a disservice to the orphan populations around the globe.
I'd like to make a comment on the standardization of the manufacturing process and the challenges that are intrinsic to that. In certain cell therapy, like MSC expansion, you can agree on a protocol, you can standardize an hematopoietic in many stem cell field. But the anomaly of islets that is escaping, which I hope will have an accelerated approval or some special consideration, is that these are microorgans. They comprise 1,000 to 2,000 cells. And if you think, you made a beautiful slide showing from small molecule to complex protein to cell, how the degree of complexity increases. Now you're dealing with 1,000 to 2,000 cells, including different endocrine cells, endothelial, mesenchymal, pericytes, and everything in this microcosmos. And you cannot standardize. So we have 1,000 pages of manufacturing report that NIH prepared for FDA on this phase 3 trial that highlights how there are ranges of manufacturing. There are different causes of death, ischemia, time, shipment. Would be like regulating liver transplantation with donorship across the country, or heart or other organ transplant. We are much closer to an organ transplant than to a standardized cell product. But what is great that emerged from this phase 3 trial is that if you meet the product release criteria, is the cells come from a pancreas of a subject that is not diabetic and producing insulin and are viable after the processing and respond with insulin to stimulation with glucose, they are likely to produce a biologic effect once transplanted. And in 90% of the cases, we met the primary endpoint at 1 year. So that has been incredibly encouraging, because it says that you don't need to overkill the standardization of a process that will never become like a standardized cell expansion of a single cell product, but will give us the opportunity to learn and facilitate application of this procedure and continue then to monitor the results and post-approval process. So in that sense, that I hope that we can have a dialogue. I'm not saying let's consider them as organ like EMA or the European or other agencies. Say let's move to a BLA, but let's try to learn from the thousands of pages of reports that are already submitted and the experience of these 10 years to see how can we move forward in the field.
Okay, thank you. So I'd like to open up the floor to any questions for the panel right now. Anybody have a question? Mark?
Yeah, just to adipose or mesenchymal, which I'm mispronouncing, already started the question. The guidance is Dr. Mark's were very clarifying in parts, but they clearly seem to express uncertainty or concern about the use of stem cells that aren't poetic in connecting the tissue, the underlying tissue, and not allowing the characteristics of those cells independently. I'm just wondering if you see a pathway there and why CBER was so conservative in that discussion.
But the best I can say there is that I think in terms of mesenchymal stem cells, first of all, because they're not made the same way by everyone, they probably are, first of all, different products and and people are making different products. People are using them for different purposes. And right now across the country, they're using them for a lot of different diseases. We'd like to make it simple enough for people to come in for, if they're not clearly accepted, make it simple enough for people to come in, have a conversation with us, get an IND, and make it not that difficult for people to get a license if they need to, so that they show that the products are effective. No one is making these into traditional small molecule pharmaceuticals, where no one's going to hold these to well-controlled randomized trials. We are very happy to license these products with phase 1-2 data if they're very compelling. And so I think we need to know that the products are what they are and that they're safe and effective for their intended use.
Any interest in, I can't help but look at the paradigm when cord blood cells were made a biological product, right? And FDA actually issued the guidance on sort of how to do it, how to rely on the same safety database and so forth. And is there any movement from CBER to maybe apply that same sort of strategy to, say, maybe adipose-derived mesenchymal stem cells, or maybe, say, a lower-hanging fruit indication like intra-articular with the knee?
So the thought has not escaped us, and it's things we're considering. I can't go into details, but I think there are places you've heard conversations about here already about things where there might be ways to try. I think what you're trying to say, are there ways that we could decrease the burden on those manufacturing and submitting a product? And we're happy to look at those, and we are thinking about them.
Yes, Diego?
So I have a philosophical question that, of course, deserves lots of discussion, but I'd just like to hear about your opinion. We know that mesenchymal stem cells are located in the perivascular space, so we can consider them parasites. When we isolate them and reintroduce them into a body, either an animal or a patient, they retake that position as perivascular cells. So if you think about transferring a tissue, yes, it's non-homologous, but if you're transferring a cell that was in the donor a parasite and the recipient a parasite, it's homologous. So, of course, these deserve more discussion, but I'd like to hear some of your thoughts about it. Especially for adipose. Especially for adipose.
Well, I think
And if you want to defer to speaking later, that's fine.
You know, it's each of these cases, I think, is pretty specific. It gets somewhat difficult to. I mean, the philosophy, I guess, is it really doing in the recipient what it was doing in the donor? Or is it in an individual? What is it doing when you put it back in? Is it doing the same thing as when you took it out? And it can be challenging, and we sit there and we have lots of discussions sometimes over this, and sometimes it's not entirely clear. We do our best here. At the end of the day, we're here. We don't. It's not like we go into a back room and sit there and say, how can we really make it difficult for people today? That's not what anyone comes to work for at FDA. We have a group of people Really, believe it or not. You might not know that from government these days, but there are those of us that come to work each day and actually really want to try to help. And we're there because we want to help get treatments that meet unmet medical needs for people out there. And we want to help facilitate that, and we just have to make sure that what we are doing, we can feel confident that there's a reasonable standard of safety and effectiveness that's met. And we understand ever more so, as has become clear as is clear in guidance and is clear in actually congressional actions, we understand that the benefit risk calculation will change depending on what disease you're dealing with, depending on what therapies people have had previously. So we're committed to making that happen. It's just that it sometimes may seem arbitrary, but what we're doing is doing our best to work our way through making sure that the products that are out there are safe and effective.
Thank you. So we just have a few minutes left, and so yes, please.
Can I raise a follow-up question to Diego Correa's question? Is the fact that depending also how you see the challenge, there may be different attitude on how you regulate. Because you could say, well, we can get MSC from the blood, from the bone marrow, from the adipose tissue, but many people prefer the adipose tissue now because the frequency is much higher than in bone marrow. And then you can think about selection, subpopulation, and everything. But the fact is that MSC are in the blood, are in the bone marrow, and in the adipose. If you extract them as single cells, shouldn't be more dangerous if you use them in an intrarenal injection, like to treat the Mesoamerican nephropathy, like you're doing in Nicaragua, rather to prevent deterioration of kidney function in diabetes, like we will do with IND, like we will apply with the proper INDs. But I'm saying the concept, maybe you can say, like you have the opposite standing in Europe. I just learned our collaborators are moving forward with encapsulated islets in alginate capsule without an IND because their EMA equivalent, FDA equivalent says, no, alginate is an inert material from algae, so we don't consider it a device, and islets are already approved as organs, so please you go ahead. Well, if we would have to do encapsulated islets here, like, would be a...
Thank you.
So actually, I have a follow-up issue, Peter, which is one of the things that's missing in the MSC field, certainly, is a registry of outcomes, because there are the use of MSC or fat or MSC-like products are widespread amongst, at least the advertising on the web is widespread. Whether they're really dealing with MSCs is another story. But the use of MSCs in terms of outcome, that it seems to me should be standardized. So if you have an osteoarthritic knee, the outcome parameters are pretty restricted and can be standardized. And it seems to me that the public would be well served if NIH and the FDA could come to terms with such a registry of outcome criteria, and the burden, the financial burden, would be on government, which is different than the current financial burden for outcome criteria, which is in private sector. But it seems to me this is something that, since we are regulated by government, quite reasonably so, that government should set criteria for use of biological products and make that widely known, and that in the end becomes the standard of medical care. And so there are a number of places where that standard can be imposed. The government chooses to allow the licensing societies of individual states, which the federal court has put down quite dramatically and firmly, that even though the physician is licensed by the state, the FDA, because everything is involved in interstate commerce, the FDA has jurisdiction, in essence, on the practice of medicine. So if you take that federal decision to the extreme, which is what I'm doing, it seems to me the onus could be on the FDA and on the NIH and on the federal government to help us establish outcome standards for certain medical indications. And osteoarthritis, putting fat or MSCs or whatever in somebody's osteoarthritic knee, although it's one of your not life-threatening activities, right? The quality of life of an osteoarthritic knee will control depression, control a huge number of patients. So we in the industry realize there is no registry. We in the industry, academics and corporations, there's 4 or 5 registries that have come up to try to fulfill this need, but it seems to me this is part of the regulatory apparatus, and I wonder what your reaction to that suggestion might be.
So I think there are various ways. First of all, I think it's a great suggestion to consider. I think it's something we can bring back to talk with NIH to see whether they're willing to fund. So I'm always happy to hear those suggestions. But I also have to say, what this goes back to, too, is we probably should also think about trial designs that would make it so that you don't need to get to the need for that registry. Because given what I hear people are seeing in terms of beneficial effects of some of these therapies, we should be able to work with you with, be they re-randomization designs from non-responders to et cetera, to find designs where the number of patients that need to be enrolled are reasonable to get to a demonstration of some type of compelling clinical benefit. So we'd like to work with people to get there, because that's what I think this field needs, because ultimately, at the end of the day, it's not just the big problem is not just FDA approval. Because it's not, in the United States, our approval is divorced from payment. It's whether the product actually ultimately will be paid for, and if it's not paid for by insurance, it's going to be limited to a limited number of people. So we'd like to help the field. Again, my concern isn't the payment issue of it, but I think that ultimately, if we don't keep in mind that there has to be a level of evidence that when the medical director of a large company, the color of my suit, or a little lighter than my suit, would be saying they want to see evidence that the thing is going to work, that there's something there. Because even if we approve it otherwise, they may not make it available to their members.
Dr. Atala, did you have a comment?
Yeah, it's interesting. I was actually going to point out the exact same thing Dr. Marks just said. I think we're, as a field, we have to temper and balance the challenges that we have. I mean, when we take a blood pressure pill, we're not thinking, is this pill going to actually work when I go to sleep, right? Because we know that's been tried. We know that it's the same pill, the same chemical or small molecule every single time. We're talking about cells, and the challenge with cells is that, they're different. There's batch-to-batch variability. The number of passages affects its potency. And not only that, the patients themselves and their genetic makeup is going to influence how the patient reacts to the treatment. So unless we really do rigorous studies with same materials over and over again that will give us the outcomes we want, we're not going to have the efficacy that we need. And it could be approved because it's safe, but at the end of the day, we're not going to have a product that's going to help the patient. And at the end of the day, really, our interest is to help the patient. So I think we have to balance a lot of these discussions today in terms of making sure that we as a field are responsible to make sure that we get together and agree upon what the standards are that we need to follow on the therapies that we need to pursue. And I think that would be critical.
Thank you. So I know we're running a little late, but I'd just like to leave this with a word from each one to say what you're excited about in the field as we're seeing it move forward. So Dr. Ricordi, can you start?
I'm excited that thanks to the bipartisan initiative and 21st Century Cure and similar strategies, we are now at the verge where we can try to understand and promote facilitated and accelerated path to testing new therapies for which there is no other alternative. And then we can learn. I like the registry part of the equation because I think mandatory reporting of every single patient should be a must. We shouldn't allow clinics or any entity to do selective reporting of the patients that are going well and forget the 60% maybe that are not working. So that is an advantage of mandatory registry. So I'm excited of the fact that we can accelerate cures, accelerate pathways, accelerate approval, but also be very strict as a field to monitor and don't allow anybody to escape mandatory reporting.
Thank you. Dr. Atala?
I'm excited about the future of standards for the field, that I think the field is really coming together to make that a reality for the future.
Thank you. Dr. Caplan?
I think we have to more honestly and completely share information. I don't think we do a good job. I think that if someone has a procedure which works, that procedure should be standardized and everybody use that procedure until somebody shows there's something better. And I think in the MSc field, the lack of standardization is criminal.
Thank you. Dr. Marks?
So I would just say I really am excited, looking forward to, I think as regulators, we really look forward to collaborating with those working in the field to bring advances to patients. And I think that's really exciting to me. We're on the cusp where we really can work together to see these advances in stem cell therapy, in regenerative medicine, actually bring benefit to people. Thanks.
Many thanks to the panel members for their thoughtful, informative answers, and have a good evening. Thank you.